moving on to our final act of the day. We will have now Suzanne Anker, that was so kind to join us during the week and today here at this event. Suzanne is a visual artist, also chair of Fine Arts Department at the School of Visual Arts in New York. Suzanne has been a pioneer of bioart for some time now. You don't need to be more explicit. Basically, Suzanne has been developing, practicing this interface between art and biological sciences by using metaphors from the biological sciences, and also some biological materials as media for her work. The work is very impressive, as you will see, and I think it's a great example of how we can thrive when you deal at the interface between these two areas. So I will shut up myself and
okay, well, thank you for being here. Thanks everybody for staying late. And I just want to let you know that I'm 1% Neanderthal, and I think that you all probably have so too. I'm going to pick up on some of the things that were talked about earlier, in the sense that art and science have different ways of knowing the world. And the idea of epistemology and producing knowledge usually happens in an experimental system, of which science is one and art is also another one. So as you gather information about those things you don't know and put them together with things you do know, you come up with some form of knowledge. This is an image of my bioart laboratory that I started in a fine arts department in 2010. It is one of the first bioart laboratories in America, where our art students use the tools of science to make art. So we don't operate by the same kinds of scientific methods, but we do absorb science as we need to for particular projects. So as you can see here, it's not a typical scientific laboratory. It doesn't have an electron microscope. We can't really afford one. But it has plants, it has coral, it has a tarantula, it has stick bugs, it has carnivorous plants, and it has molecular equipment. We also have microscopes, we do electrophoresis, we test for DNA in GMOs, we do tissue cloning of plants, and we raise questions about altering nature, either in the laboratory or outside of the laboratory. And in most art courses, you never get into a philosophical discussion such as that. That the discussion is rather much more about comparisons of space and texture and line, etc. But it is my understanding that a work of art is a proposition. It's a proposition about what you see the world, how you see the world, and it's about world making. And when I became chair of the Fine Arts Department 30 years ago, I went through the course catalog and crossed out every word that said expression in it, okay? Because art that is about expression makes no sense, because it's about you anyway. It's about intersubjectivity, which you are the vehicle for delivering. And so I like to think of art as both a conceptual process and an emotional one that are tied together to form a proposition.
A new generation of biohackers is tinkering with DNA to create plants with new colors. At the School of Visual Arts in Manhattan, plant researcher Sebastian Cachopo is trying to create genetically modified plants, mostly from information he learned on the internet. One of his pursuits in the school's bioart lab, the blue rose, considered the holy grail of horticulture.
I think the main reason why people are trying to make blue plants is because there are very few, if not any, blue plants out there that are manipulatable or growable in a specific condition. Basically because those high saturation colors just simply do not exist in nature.
GMOs, or genetically modified organisms, have drawn a considerable amount of controversy, with concerns about the potential impact on health and the environment. Cachopo doesn't think such worries apply to projects like his.
The main argument I would have towards anybody who contests us going through with this project is the fact that it'll be contained and it's a plant. So we're not working with pathogens or any other type of human or mammalian tissue.
Suzanne Anker sees the process as more of an art project than a science experiment.
Artists here use science as a tool, so they are not engaged in research in the same sense that a scientist would be. Blue is kind of an area of wonder. Children say, why is the sky blue? All the time. That's one of their first questions. And I think that is in part the background of this. But I do think that even if it doesn't work, it doesn't matter because it's art.
The process is meticulous and complex. It all comes down to identifying and manipulating a specific gene that could give the plant its color.
The gene in question, called AE blue, produces a beautiful cerulean hue in bacteria. It's never been tested in plants as far as I know. So this will be the first time that we actually try to put it into a plant.
With the project about halfway done, Cachopo is still tackling problems, but he says even if it fails, the experiment won't be a loss.
In an ideal situation, in another 3 months, we'll have our blue plant. In the worst case, it might not work at all, and that's the risk that we're willing to take, and that's the risk that we want to attempt, because even a failure might be useful in terms of understanding the processes at hand.
This was on the front page of the Wall Street Journal, which never happens in an art school. This is in their archive. They did a little video as well. But I want to clarify the remark I made, meaning if it doesn't work, it doesn't matter because it's art. Now, it sounds like a very snide remark, but it's not. And that's because, in my opinion, for every scientific discovery, there's a unique and discrete and parallel one in art. And let's take the example of the blue flower. We go back to Novalis, one of the early poet and philosophers, the late 18th century, in which he wrote about the blue flower as a symbol of the ethereal, of the infinite, of longing, of the ideal. And that symbol continues today in the films of Kubrick and David Lynch, in which the characters are wearing blue flowers. There are many other examples. Tennessee Williams' play is about the fragility of Laura. So that symbol sets parallel scientific sets and continue to develop and kind of produce knowledge. Now, the scientific community has tried to create a blue flower, blue rose. Actually, Florigene is the company that had tried it, and because of the complex chemical pathways and the pH levels, etc., they came out with a lavender rose. Okay, so it is still one of those unattainable kinds of quests, and whoever can come up with a blue rose will essentially become a billionaire. If you go on Amazon.com and look for blue rose seeds, you can buy some, but in this age of misinformation, there are none. Okay? So now the question of bioart, which is a very hard concept to get across because it's almost an oxymoron. And when I went up to the president of my school and said I wanted to make a bioart lab, he had no idea what I was talking about, okay? I had just moved into a 54,000 square foot building with all new technologies, et cetera, that I had put together. And he let me do it, okay? And now it's been a very big success. When parents come and they see the lab, they think that this is a real college, not just an art school. And they're very taken by this as a way of understanding the world. So now he's very happy, and he knows what bioart is. Now I'm going to try to explain it to you. I wrote the definition of bioart for the Oxford Dictionary of Art, so I'll have to stand by my words. It's a contested term, and no one really knows where it came from. Certain people say that they coined it, and then of course that becomes repeated in the press and re-repeated, etc. But it's still unclear. But I look at it as something in three genres. One is wetware, which happens in the lab, which is why I wanted a lab. And another is computer technology, which is part of the instrument, is part of the process. And the third area is any kind of medium, painting, sculpture, text, film, etc., that points to the direction of altering nature. So here we have Mark Quinn in this genomic portrait of Sir John Sulston, very famous geneticist, in which he took cultured some samples of him and put it in this stainless steel frame. And when you look at this piece of paper that looks like it has been foxed, you see yourself. So Mark's idea here was that you and your DNA are a reflection of each other. Now, I don't think that's true, but I think what's interesting about this piece is it's the first abstract portrait to enter into the portrait gallery in London. Okay? Second is my work, remote sensing, which is done with rapid prototype computer technology that transitions two-dimensional objects into three-dimensional objects, and I'll be showing you a lot of that. And third is Patricia Piccinini, who is an Australian artist. This is a silicon sculpture of a projected hybrid between humans and dogs. And the question here that is raised is, how human does something have to be in order to have the rights and privileges of human beings? This was a conversation we had with gorillas a number of years ago. And what bioart does that science doesn't do is it captures the cultural imaginary. It is about the fears and hopes and promises about the ways in which science can benefit society. And what's ironic about that is that in popular culture, both the artist and the scientist are presented as mad persons that sort of live in some sort of little hovel attached to some idea and are overly consumed by that. So that is sort of the popular tale here that may bring them together. So let's go on, and I'm going to show you a few pieces of my work that show a process of growing, because ideas grow besides materials. Not materials, but living things. And this living thing here, Astroculture in 2009, I grew vegetables in a gallery, and using LED lights that had the red and blue lights that would create photosynthesis in the plants. And this was part of my idea about the way that agriculture is moving indoors because of the lack of pollinators, as well as climate change. So as we proceed, we are going to have to think about new ways of producing food. And if you look at the pictures, and I would go there every few days and take some different pictures, they're all fuchsia. And so it got me to believe that fuchsia is the new green. And here you could see a little sprout. All right, a few years later, 2015, I got invited to be in a show at Cathedral of St. John the Divine, an unfinished Gothic cathedral in New York City that's magnificent. And rather than having 3 bins on the wall, they gave me a whole chapel. So I said, all right, I have to now expand this piece. How am I going to do that? So I figured out a way to construct 31 of these boxes, which are off-the-shelf boxes. Everything that's in this piece is off the shelf, okay? And if you think about coincidence, the boxes were able to hold the 24-inch LED lights, and I didn't have to do anything other than connect the electricity, okay? Work smarter, not harder, is the trick. And people from out of town visit this church regularly, and they didn't believe this was real, okay, because they never saw anything like this. So school children would come, and the priests would have a conversation around this piece, but essentially, if you were able to shine your phone light on it, you would see that it is actually green. And alas, what happens here? Why an artist is growing space lettuce. I used some of the same lettuce seeds that the astronauts used to go up in space, and this was a piece that was written by the Biodesign Challenge, who are an entity that have competitions about bio-art and bio-design every year. And then I get an email from a salad magazine in Italy wanting to know how to grow these lettuces, okay? So when you put something out there, you don't know what the response is going to be and who's going to pick it up. Now, of course, this idea is from NASA, Veggie 01, the experiment on the International Space Station. And you can see the fuchsia color there. And these astronauts were allowed to eat the lettuce. A few years earlier, some Japanese astronauts went up and they grew cucumbers, but they were not allowed to eat the cucumbers. I think that what's interesting about outer space and botany and food and minerals is that's the next frontier as well. If you boil water in outer space, you get one big bubble. If you plant a seed in outer space, you get 2 sprouts. So we are just on the edge of a new paradigm cliff, and I'm looking forward to seeing what astrobiologists come up with. And here you can see they had hydroponics, which was a very good system, although I had my assistants go up to the church and water plants. This is a piece by Jalila Asadi, which I think is a terrific piece. She worked with some scientists and silkworms to try to combine human cells with the spider silk to create bulletproof skin. And although it didn't work, it almost worked, which means that with the right resources and continued experiment, it might work one day. Now, this is probably a work that would never get funded by people who write grants, but this is what an artist can think of, okay? And I think it's excellent. Now, Heather Dewey Hagborg came up several times in our presentations. She's visited the BioArt Lab as well. And what I've noticed about some of the practitioners in bioart, some of them are in biodesign, some of them are in speculative design, some of them are in bioarchitecture, that there's a continuum or spectrum with different ideas in mind. She comes from a digital background, so in terms of creating the portrait of her face, which is done on a 3D modeling program, that was an easy thing for her to do. So collecting these cigarette butts and gum in Brooklyn and bringing them to a place called Gen Space, which is a do-it-yourself bioart public space, and they are popping up all around the world. I don't know if there's one in Portugal, but they are all over the place. And she got involved in deciphering the genetics of these items and using the SNPs method, which can give you eye color, the size of the nose, the size of the lips, etc. It can't give you age, okay? She then took that information and ran it through a facial recognition program to see if she could construct an image of the person who left their DNA behind. Now, because this is bioart, there's no peer review. So whether or not this is true is still up in the air. But since art itself is a lie that tells the truth, this becomes another question of what kind of license do bioartists have to say they have created something that's scientifically valuable when in fact no one has repeated it. So I did write an article a number of years ago called "Where is the Art in BioArt?" asking the question whether is the best bioart the one, or bioartists amateur scientists, or are scientists, is the best science put on the cover of science journals, or how does that get on a science journal, okay? Probably by the use of its visual appeal, because images have power, and that kind of power, as we've just heard, is not necessarily rational. It is spontaneous or intuitive or whatever. And pictures change people's minds, okay? Another piece that we heard about was Christiana Agapagas and Sisal Tolis, who made cheese from the bodies of the synthetic aesthetics team. Now, they are both scientists. They both have PhDs in science. Sisal Tolis is a specialist in scents of smells. She has a whole collection of smells. Christina Agapagas also has her own company in Boston. So they went, as we heard before, and took some images of Daisy's armpit, a philosopher's toe, Christian's hand, Seisel's nose, and created these cheeses. Now the rumors which surround this is whether they ate him or not, but they wrote a really long paper about this. So the science was correct in this issue. The other piece we saw earlier about the woman giving birth to a platypus, that too was part of what's now called speculative design, for designers to ask questions not about solving little problems, but what if? And certainly we have seen in science the birth of closely related animals giving birth to one another. So I find that an interesting piece. Another piece that is kind of scary, and you could say these are beautiful, but they're deadly. So can a deadly item be termed beautiful? This photographer, Craig Ward, went around the subways in New York City and took swabs from the poles that you hang on to, or the hangers you hang on to when you're riding the subway. And what do we have here? We have E. coli, Salmonella, Staphylococcus, Bacillus, etc. So these are just out there with us. There's no separation. We are the environment. Okay, so what I meant to say here, in cultures of entanglement, we have microbes on us, in us, around us, and we are part of that nature's diversity. I'm going to show you a number sets of my work. These are called Vanitas in a Petri dish, which I started in 2013, based on the fact that everything living has an expiration date. And the Vanitas paintings in the 18th century, etc., were all about the fragility of life and the need for more spiritual rather than material existence. Because what happened during the Dutch Golden Ages is the mercantile class got very rich, and people were tasting foods from far away and wearing fancy clothes, etc. So I decided to update Vanitas using a synthetic arrangement. One thing from nature, one thing from culture, or more. Okay. And one of the things you can see about culture is that the colors of culture are second nature, that they simulate the colors in the natural world. So we have a sunflower, the rubber ducky, we have some ceramics, some rocks, and some flowers here. And in these, we have a cutout of a pansy and some dead flowers, some meat, and on the right-hand side, we have a shrimp with some glass in the Petri dish. Now, the Petri dish, again, is a double metaphor. We know what it is in the scientific lab, okay? It is a place to keep things sterile so that you can see what emerges. It also is a metaphor in society. And you will read in the newspapers or other journals all the time, blah, 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 is the Petri dish for blah, blah, blah. So it is about the way the unseen emerges from what is underneath the backstory, so to speak, of what's happening. So, how do you turn these vanitas, an art historical genre, into a micro-landscape, another art historical genre? Well, through creating a third axis and essentially rapid prototyping these pieces, which through rapid prototype machine uses the same data from the photograph, but then prints out one layer at a time, like a sedimentary rock, in color, the height and configuration in 3 dimensions. And this series is called Remote Sensing, and again, another allusion to NASA, because they have their satellite data, which is able to visit places that are too toxic or war-torn to visit, so that we have a camera in the sky. And here you can see the little flower piece, the rubber ducky, etc., turned into 3 dimensions. These were originally in an exhibition at the Parish Art Museum in Watermill, New York, which is like Southampton or East Hampton, if you're not familiar with that. And the little Petri dishes were in a case. And the people who came there tried to match up what was in the case with what was in the wall. And it wasn't that easy, okay? You would think same data, you know, processed differently, easy to match, not easy to match. But what I learned from this was that the machine that made these pieces taught me an aesthetic trick, okay? Because they read the data and were able to create edges around the sculpture in color that I had never thought of doing. So because it was a mistake, and someone said here that machines don't make mistakes, they make mistakes, and sometimes those are beneficial mistakes. I've been trying to reproduce this for a while, and we're not getting the results we want. Okay? And here too, the glass vessels also work as magnifying and distorting glasses that add another dimension of meaning to the pieces as we transfer information from one form to another. And here are some examples. And I said that living things grow. Well, so do ideas. So a few years later, I was asked to do the solo exhibition at the Everson Museum in New York, the first American museum by I.M. Pei, and I upscaled everything here. So instead of 24 images that were about 2 feet by 2 feet, there are 48 images that are 44 by 44. And you can see the scale of this here. And again, the same kind of game of looking at the wall and looking at the images are in play. This giant cube is called a Frank case, if anyone ever is looking for a ready-made giant plexiglass cube. Okay, and again, the reflections are part of the piece. All right, what happened in 2022? I get a call from China who invite me to go and participate in the Beijing Art and Technology Biennial named Synthetic Ecology. So here I am with my vanitas that are talking about the mix and match of the natural and unnatural. We could even talk about that in terms of genetic engineering, and all of a sudden there's a whole show at Q. So I decided I would send them the work that they requested, and then they came back to me and they said, we want more, okay? So what started out as this little installation all of a sudden became this enveloped kind of situation, and of course I was very happy about that. But I also saw a lot of the other work in the show. I did not go to Beijing, even though I've been there many times, and a very, very interesting work in bioart and other forms too. Okay, another piece of mine, which is propositionally based, and these are the series of biota. These are handmade porcelain sculptures of sea sponges, but they look like dead coral. And I'm thinking about these sea sponges that I collected and didn't want to use them in their natural form, because then they would just look like a bunch of sponges. So I figured I'm going to transform these materially. All right, I tried to cast them, didn't work. I tried to dye them and didn't work. I tried to spray paint them, didn't work. I tried to use my laser, didn't work. I said, there's gotta be a way that this is gonna work. And I ran into a colleague of mine, and he said, did you ever try a burnout technique in ceramics? I'd never worked with ceramics. I said no. Okay, so what I did is I took these sponges and dipped them into liquid porcelain, let it dry, dipped it again, let it dry, blew out some air bubbles, put it in the kiln, and what the heat in the kiln did, it evaporated the sponge and left the image in porcelain. So this is ghost of sponge, actually. This is the negative of what was there. So once I figured out how to do this, of course, I decided that I would continue with these, and made about 150 of them, and each one is unique. So there are different types of sponges. So I originally had this idea. I was curating a show in Istanbul called Fundamentally Human, Neuroscience and Visual Art. And I was thinking, well, what would be a good way to represent neuroscience? And that's where I came up with the sponge, because the sponge is like is a filtering system, and the sponge also takes in everything around it, like the brain. And there are even some sponges or some coral that look like brain coral, etc., with the same kinds of configurations. So, so I put a small version of this into the show at the Para Museum, and everyone said, well, that looks like dead coral. And I said, yeah, it does, but do you know what's happening with coral reefs lately? And they're in decline, and so one thing we have here is we have the sponge's brain, and then we have sponge as coral, which is dying, and this is one of the attributes of art. It can mean more than one thing at the same time, that it can hold a contradiction. And I added some little figurines and kind of little neurons in here because I was reading about changes in the brain through time, and the way in which art was part of that development in culture. So there's a little Venus of Willendorf's in here as well, just to talk about art history and bring it back to art history. And my background is in art, but I was always a non-technical artist. I made sculpture and I made photography, which I still do, and drawings. But unlike other bioartists, I didn't come out of tech. Rachel Carson, she came up again today, huh? And this is Silent Spring, which had an amazing effect on American thoughts about ecology. The name of the book refers to the fact that DDT was being used as a pesticide, which she called an ecocide, and that the robin's nests, the eggs in the robin's nest, were too thin to hatch chicks, so that the spring would be silent. And she was ostracized in the community. She was a woman at that time, and she also did not have a PhD. She only had a master's degree. But eventually, things came around, and she was rewarded for her efforts. But I'll show you this little clip.
Ms. Carson maintains that the balance of nature is a major force in the survival of man, whereas the modern chemist, the modern biologist, the modern scientist believes that man is steadily controlling nature.
Now, to these people, apparently the balance of nature was something that was repealed as soon as man came on the scene. Well, you might just as well assume that you could repeal the law of gravity. The balance of nature is built of a series of interrelationships between living things and between living things and their environment. You can't just step in with some brute force and change one thing without changing many others. Now, this doesn't mean, of course, that we must never interfere, that we must not attempt to tilt that balance of nature in our favor. But unless we do bring these chemicals under better control, we are certainly headed for disaster.
What you see being sprayed into the swimming pool, DDT. And that sort of set off, this book was well received by the visual arts community and the culture at large, actually. This was the tune in, turn on, drop out time. And Back to the Earth, the whole Earth catalog, and the introduction of performance art and video art, and land art, sort of trying to redefine the limits of art. And there's a lot of seminal work being done in this era. And President Nixon signs the Endangered Species Act in 1973, which has now been neutered practically by the administrations that came after him. This kind of came about when the American eagle, the symbol of America, was in danger of extinction, and we certainly had to do something then. Which just brings me to a side thought about a Robert Rauschenberg painting or combine in which he had an eagle hanging off the edge of the canvas, and it was owned by Ilyana Sonnabend, which was a very big-time gallery in New York. Her husband was Leo Castelli, and when the Sonnabend Foundation or whomever tried to sell the piece, they couldn't sell it because it had an endangered species on it. So I thought that was a very interesting story. And of course, Earth Day, 1970, look at all the people in the street. And I'm going to show you one more simple body of work of mine quickly. There are 80 pieces in this series, but I'll just show you 3. During COVID and under lockdown, I was living in East Hampton, which is on Long Island, because the city was sort of, New York City was locked down, and I was directing my courses online and running my department online. But I had this really fantastic garden that I grew. And so I started taking pictures of everything I grew because I like to look at how the garden is always in flux. You can't walk into the same garden every day, okay? And as I started taking these pictures, they turned into collages, because I'd go outside and I'd see my sassafras tree in November with buds. And I didn't know whether to put a coat on or to wear shorts. So I came up with this idea, how am I going to talk about climate change? I'm going to talk about climate change by creating mono-seasons, where more than one season actually happens at the same time. So here are some examples of these works. These were done very traditionally with cut and paste, with a scissor and glue. Then they're rephotographed, and then they're printed large scale. And this one is called Arctic Garden, and you know that the Arctic was once tropical. Okay? So this is sort of the geological surround of the Anthropocene. Here is another one, snow petals. And this one, pyrexia, which means fever, is an indication that we're also in the Pyrocene, okay? That all of the fires that are prevalent in Australia, California, etc., are with us under these new climate conditions, where strawberries grow in Greenland, and and permafrost is melting. So I thought this was a good way to show something about climate change. I mean, what artists do is they take, well, not artists, all artists, this artist takes an idea and then tries to figure out a way to visualize it. And it's not that easy to do, okay? It's not about expression, it's about conception. And you might call it hypotheses, that this is what I want to achieve. And then you try this, you try that, like I did with the sea sponges, until you get something that meets your standards. Because, you know, one has to edit their work as well. And for the last two images I'm going to show you, these are two collaborative projects that I did with my students. And we entered into two competitions, one called iGEM, which is a big biotechnology competition of schools in Boston every year, and the other was the Biodesign Challenge. And our first project was called Soiled, and what we decided to do was collect soil samples from all 5 boroughs in New York. And as you can see, the graphics here are part of the image. It's made out of soil, and we're very precise about what we were doing. And we would locate these different gardens. We first of all asked everyone at my school if they had a garden or access to land, and then they asked their friends, and we got all of these people. And this is a typical urban garden in Brooklyn, New York, and people who live here may plant vegetables and eat them, and they have no idea what's in the soil. Now, originally we thought we would test for toxins, but then we thought we'd probably get sued, so we just tested for elements. And what kind of struck us was the different pH colors that are very much like a color chart that one uses in a painting class. And we used the Lamont system, which agriculturalists use. And here you could see the little color aid pieces. And we would use soil and put this chemical liquid in to extract it, and tried to match up the color with these charts. We collaborated with a do-it-yourself scientist, and he had the idea of trying to create a microfluidic chip. So we used this copper board, and as a positive relief, we changed the hydrophobicity of it. We created actually a maquette of the landscape using these LED lights. And this was a sketch that our students did, diagrams of the installation. For the home gardener to test their soil samples. We never followed through on this. You know, we don't have the manpower, nor do we have the intellectual property rights that would take to do this. But it's a very good idea, you see. And we had another good idea a couple of years later. But let me finish showing you this. This was our installation at the iGEM Jamboree, and everybody thought we were a company because we were well-equipped, designed, we had postcards, we had everything a company would have, but we were a school. And then later on, we actually created these charts based on our findings. And you can see here that aluminum is very large on the right-hand side and very small on the left-hand side. These are two different plots of land. And the same process was used in each case of labeling. It was consistent. Now, getting artists to work together is like herding cats, okay? So, because usually when you teach an artist or art student something, first they try to do it the right way, and then 5 minutes later, they're making up their own rules, okay? So this was a situation where they actually paid attention. And if you put all of these in together, you look like you have Damien Hirst paintings, okay? But this is what uses color as an identity marker of a particular element. So the visualization of color crossing over this way is also very important. Hence, and behold, we won the gold medal for the Best Art and Design Project, an iGEM. We were also nominated with MIT, Harvard, and other schools for Best Integrated Human Practices, Best Education and Public Engagement, Best Presentation. So we were very happy with that. Okay? So that was in 2015. In 2016, we had another idea, and this time for the Biodesign Challenge. And we decided that we would genetically engineer a tomato, a beefsteak tomato, so that it would have the proteins that beef has. So you can see in this poster of ours, we have a tomato, and we have a meatball in the shape of a tomato. We've crossed out the cow. We have our little seedlings here. We have some products, etc. And here we can see tomato culture in vitro. And what struck me about the tomato was the tomato's history. And the tomato comes from Mesoamerica, and it was once very small. And then it went to Italy. Didn't come to the United States because everyone thought that the tomato was poisonous. And there's even rumors going around that someone tried to kill George Washington by putting tomatoes in his salad. So every part of the tomato plant except the tomato is poisonous. But look at this correspondence between the Aztec calendar and the tomato slice. I thought that was very interesting. And here's the science behind it, which of course was led by our do-it-yourself scientist. And here we used the agrobacteria. We induced and grew the baby plants, and we actually got a tomato. We actually grew big plants. We got the tomatoes, and we knew that the gene was in doing electrophoresis. Okay? And as part of our presentation, we made tomato products because you have to bring it back to art. Okay, so here's the Campbell soup can as tomato, and here's the final.
BioArt Lab, where we've been designing the future of food for over 30 years. Good. This laboratory, the first of its kind, is where biologists and artists work together tackling some of the world's biggest biological concerns. Over the years, we've spawned some unique results, but nothing as successful, impactful, and delicious as this. Meet the tomato, the amazing protein-packed fruit that started it all. A biological breakthrough that changed the way we think about protein. I have something amazing to show you. Randy, get the thing.
As you wish.
Before the tomato, humans relied heavily on animals for their daily source of protein. With a growing global population and a strained agricultural system, we recognized animal protein was not an ecologically sustainable source. There had to be an alternative method. We put together some of the brightest scientific and artistic minds we could find, and the result was impressive. Take a look.
This only hurt a little bit, JK.
2016, the BioArt team at the School of Visual Arts engineered the world's first fruit to produce the same protein, myoglobin, found in animals, the myotomato. They found plants to be deficient in megadoses of protein necessary for a nutritional human diet. So they translated a DNA sequence found in beef and inserted it into a tomato's genome. The result? One little tomato possessed the same amount of protein found in an 8-ounce steak. This breakthrough creation caused quite the stir. It seems not everyone was a fan of this amazing scientific achievement.
GMOs are dumb and stupid and poison.
Of course, we're not happy.
However, in the year 2030, mad cow disease almost completely wiped out the cattle industry, creating an urgent need for the myotomato's amazing protein power. And soon, hearts, minds, and mouths were open. The myotomato ushered in a global movement that ultimately ended our need for animal consumption. Not bad for a group of art students, if you ask me.
That's it. Thank you. Had a very talented crew because it's an art school. They knew all of these different techniques, video, rapid prototyping, you know, these holograms, whatever. So they did a great job. Any questions?
Did they eat them? Did they eat the tomatoes?
No, we did not eat it. We have a rule in the lab that whatever is in the lab is not food, okay? So we did slice it up and freeze it, so we have samples of it in our freezer.
So you have not falsified the hypothesis that they could be poisonous.
No, I haven't. We brought in another scientist the following year to see if we could carry it further, and she took it a little further as well. But there are no PhDs at my school, and we didn't have the scientists in order to really formulate this. Now, this was before the Impossible Burger, okay? This started out as a speculative project, but it actually worked. Okay. Not to completion, but it worked well enough. Okay. Now, do you think you would get funding for a myotomato? Yeah.
I have a question about the vanitas. I always liked the old ones, like the. And you know, normally that what you also see is that you also represent the decadence a little bit, also like a dead nature. So all your vanitas, in fact, were still kind of fresh and alive. So is that like what was the idea, the decision behind that, not to have anything rotting or anything?
There were things rotting.
There
were things rotting.
okay.
We just didn't have any real close-up. We had lots of dead flowers. We had some bones.
Okay.
We had some insects, dead insects in there. We didn't have any timepieces.
Okay.
But also, I think it was a way to show the correlation of second nature of artifacts as they replicate the colors of nature. And you know, look what's happening with the albatross. They're all eating the plastic in the ocean and they're dying, you know. So there's this kind of question about our products that we manufacture and what the risk reward is, and it's certainly out of control here.
I was just thinking about the fact that obviously we didn't prepare together any kind of our presentations, and there were quite a few similarities. And there is one basic similarity in all this. My first close encounter with science and art was the works of, ages ago, works of Nat Kahn at the Exploratorium in San Francisco, and they were about physics.
That's right.
Now, the whole day what I've been talking, it's not exactly science and art, but actually life sciences and art.
That is correct.
Should we read something into that? I mean, it's because this is science now, the problems are there?
Yeah, because in the last century, physics was the hard science, and the biological sciences were considered soft. And in the 21st century, the biological sciences are the key sciences now to open up innovation and experimentation. And so the paradigm has shifted. It's no longer the atom. It's whatever it is. I can't tell you. It was the gene for a while. Now it seems, and that's why the term bioart is different than art and science, okay? Because art and science has been around a long time. Georgie Kepish at MIT, the Xerox Corporation with Billy Kluver would send artists out to California to work with technicians, but the biological sciences are new, and the molecular level is new in art. But it follows the trajectory of Romanticism, actually, and that's why I sort of mentioned Novalis as well, and Alexander von Humboldt as well, who talks about nature as something one can experience empirically, but also calculate. So I think he had it correct, and I think that experiential knowledge is as important as, I don't know, calculated knowledge.
If I can. So do your work or your projects have to be submitted to some kind of ethics committee, or, because I'm thinking about the green fluorescent rabbits, right? So are there any limits for what you can?
Yes, first of all, we have a BSL-1 laboratory, so we don't deal with anything, no blood, no sperm, no, no viruses. So we deal with K12 E. coli, which is neutered, and several other things that are pretty harmless. Yes, there are ethical standards, but we don't do anything that requires that. Now, the bunny, and I've discussed this with Rui, and we have to get we have to ask one of the fluorescent guys, okay? Because the bunny, no one ever saw the bunny, okay? No empirical evidence this bunny existed. The story is that they didn't let him take him out of the lab, out of the lab. Well, you wouldn't see a full-grown bunny glow green with that kind of fur, okay? And in my opinion, and in others, it's a Photoshop creation, okay? But even if it is, it's again a symbol. It's a critique of altering nature. So it has value, not scientific value, because I'm not sure you can even do that scientifically, but it has cultural value and caused an uproar. Okay?
don't want to. I have a question which sounds a little bit out of the blue, but it was stimulated by the fact that you're talking about the vanitas, then you had the food, and you had the tomato. So I wonder many times why is not gastronomy included in the arts? Because it seems to me that would have all the requirements for that. It surprises our senses. It is also the aesthetic component and so on. But it doesn't.
It is.
It is.
It
now.
is. We teach a section on molecular cuisine, and we have used those techniques like spherification to encapsulate artificial blood, sweat, and tears that then were in a project. And also, I forgot his name, the famous Spanish molecular cuisinist. He had a show at the Drawing Center in New York of his drawings of plating. And yes, food is part of science, and it's part of aesthetics, and it's a complete sensual experience. So yes, and I have another course I started called Food in the Era of Climate Change, because our whole food system is now changing. The Georgia peach, which originally came from China, can no longer be grown in Georgia because it's too hot. Okay? So their symbol is now crossed out.
Okay, so there are no further questions. Thank you again for